If you're here to learn a bit more about what a gene is and how it works, then welcome to Genetimix. My name is Tim, and on this channel we cover genetics and a mix of other bits and bobs including science, communication and critical thinking. We covered an introduction to DNA in a previous video, so in this one we're going to concentrate on what a gene is. And it's a surprisingly complicated topic. So much so that, right up top, a gene has two separate definitions. The first is to describe a gene as the unit of heredity, one of the items that will be inherited by the next generation. You can also describe a gene as a blueprint or template for a product, which is usually a protein, but which can also be RNA. I will start with the second of these definitions and then come back to the first. What I've decided to do in this series is to layer on a bit more detail in each video as we go along, so we will often start with an extremely simplified concept that we can build upon as our understanding grows. In the last episode, we had a look at how stretches of the DNA on each chromosome can form genes, and also had a look at the basics of gene expression, where DNA is translated first into RNA. When this RNA is a template for a protein, it's called mRNA, which is short for messenger RNA. Humans have around 30,000 genes. This particular illustration of a gene is again highly simplified. We will expand on this as we progress through this series. Each of our genes consists of stretches of coding DNA that codes a product, called exons, interspersed with regions of non-coding DNA called introns. The exon-intron boundaries along here are marked by DNA motifs, and the introns are spliced out of the mRNA transcripts before translation takes place. Some genes have splice alternatives, allowing different products, also called isoforms, to be produced from a single gene. So in this very simplified example, we could have exon 1 over here being paired with just exon 3 to produce one protein, and then exons 1, 2 and 3 together, forming a second protein product. An example in humans is the CD44 gene, for which at least 29 known protein variants have definitely been detected, although some studies suggest that there could be many more. The current record holder is the Drosophila DSCAM gene, for which there are over 38,000 known protein isoforms. Drosophila melanogaster is the species name for the fruit fly, which is best known for crawling all over your fruit and being found drowned in your glass of wine. Drosophila is a model organism, a non-human species that is usually easy to breed in a lab setting and which is used in research to help us understand specific areas of biology. Other familiar model organisms include the bacterium E. coli and Mus musculus, the mighty mouse. Getting back to protein products, on top of all this, the final protein products themselves can have multiple functions as well. There are so many of these that a separate database called Multifaceted ProtDB has been put together that pulls in information from other data sources to help scientists better understand these complex proteins. One example is the CFTR gene, which stands for Cystic Fibrosis Transmembrane Conductance Regulator. The CFTR protein can act as a chloride channel in the cell, but can also regulate the activity of other sodium and chloride channels. We have two physical copies of most of our genes, one on each chromosome, and often these copies are not identical. These alternative versions are called alleles, which are functionally slightly different versions of a protein. If you inherit two defective CFTR alleles, one from each parent, you will be born with cystic fibrosis, a disease that, among other symptoms, inhibits mucus production and makes it difficult to breathe. CFTR is interesting because the presence of a single defective allele is silent. You can be a carrier of cystic fibrosis without ever realizing it. The defective CFTR alleles are among many common disease-causing mutations that can be screened for using genetic testing in early pregnancy. Some genes are so complex that we even refer to them as a locus or loci. In some cases, a locus may be entirely absent in some individuals. Another straightforward example of alleles is the blood group gene, which is helpfully called ABO. Humans have two copies of ABO, one on each chromosome 9, and each allele, or gene copy, will be either an A allele, a B allele, or an O allele. If you are AA or AO, you have blood group A. The same with BB or BO for blood group B. The other options are AB blood group or OO for blood group O. The O blood group allele is what is known as recessive. It's basically silent unless only O alleles are present. It will be drowned out by the dominant A or B allele if present. As always, this is actually a little more complex if you dig a bit deeper, but it serves nicely as a start for 10. At the other end of the scale is the HLA gene family, which has tens of thousands of alleles, and which I will be covering in a future video. The genome is defined as all the genes or genetic material present in a cell or organism. Only a small fraction of the human genome is transcribed and then translated into a functional product. A lot of the genome consists of pseudogenes, which may be transcribed, but not into anything useful. Some alleles are null alleles, which are translated all the way into proteins that are no longer functional. The genome also contains remnants of viral infections, because retroviruses have had their sequences copied into our DNA at some point in our ancestry. These sequences are called endogenous retroviruses, or ERVs. Endogenous here meaning originating from within the organism, because they are now part of our genome. We'll cover ERVs and viruses in a lot more detail in a future video. Gene expression is also a very complex topic. There are whole families of genes that exist only to help control the function of, the expression of, other genes, and there are many environmental factors that also affect gene expression. Again, we'll have a closer look at this in the future. As you can see, it can be difficult to see genes as discrete units, which also complicates our second definition of a gene as the unit of heredity. Heredity being defined as the biological processes where traits and characteristics are passed from parents to their offspring through genes. In the DNA introduction, we discussed chromosomes, where the genes live, and meiosis, the process that creates sex cells, each with half the chromosomes present. Part of the process included in meiosis is called recombination, or crossover, where pairs of chromosomes can swap parts with each other. This makes each sperm and egg cell unique, and is the reason why we cannot consider chromosomes to be the main unit of inheritance. We had to find a subcomponent of the chromosome, and what fits best is the gene. All this feeds into the essence of why genetics is so interesting, but also illustrates quite well why concrete definitions in biology can be challenging. Biology is messy. This will not be the last time that this phrase comes up. I will also be doing a deeper dive into the field of genetics, where I'll dig down into heredity, start taking a look at genetic variation, and review the history and breadth of the field. The sequencing of DNA and the analysis of genetic data using bioinformatics are also planned for upcoming episodes, so stay tuned for these. And that wraps us up for today. Don't forget to like, subscribe and comment. My social media feeds and Patreon are in the description below. Remember, science is amazing. Thanks for joining me, and I'll see you again soon.